Hello, everyone. This is Purnima. I'm the product manager, and I've been working with Krishagni, the company who is developing OpenSpecimens. And we'll start with some, introduction about ourselves, and then we'll dive into the demo of the application. We, as a company, have been working on OpenSpecimen and with multiple biorepositories, research centers across the globe since 2009. And we currently work with multiple research centers, universities, hospitals across the globe, about 90 plus biobanks, and across the 20 plus countries. Some of the leading research centers like Washington University in St. Louis, Johns Hopkins, Weill Cornell, Baylor, and there are others outside US as well, in Australia like UNSW, and in University of Oxford, and so on. As you see, we are working with these universities, so it is more at an enterprise level. So OpenSpecimen is used for multiple sites or biobanks within a center. So it is used at a university level. There are, of course, other clients who also use it for their specific studies, research needs, or at a smaller scale, might be just for few studies as well. So it can be used at any level. We as a company are solely focused on OpenSpecimen product. So we are a product-based company. We are the primary developers, and we also provide support or any kind of enhancements, integration, migration needs that is required for OpenSpecimen implementation. We provide services around that. We are a SOC 2 Type 2 certified company. So this is required because we handle sensitive data, so to be in order to be HIPAA compliant and so on. So now let us move ahead and look into what are the features and highlights of OpenSpecimen. First and foremost, what does OpenSpecimen do? It tracks complete life cycle of the specimen, starting from collection, processing, storage, till it is utilized or given away to the researcher. The whole process can be tracked with all its data and with all its audit and everything that is required for tracking the specimen can be done in OpenSpecimen. So some of the workflows that can be done is starting from when you collect the sample from the patient. So you enter the patient demographics, clinical data, consent the participants, and once the patient is registered, the collection or accessioning of the specimens and sending it to the biobank or labs where it is received and processed and stored. And in order to do all this, the additional data that you need to capture, for example, centrifugation details, freeze details, all these can be captured in OpenSpecimen. And once you have all this data, you can make it accessible to your researchers who can log in and search for the specimens of interest and request, which you can finally process and distribute. So this whole process can be tracked in OpenSpecimen. Of course, none of these are mandatory. So we'll be seeing some of the use cases in the demonstration. You can skip certain things. For example, a lot of our clients use different applications for consent data and patient data. It might be REDCap, EPIC, and so on. So the first and second step might be done somewhere else. And once you come, once the specimen data comes into picture, that's when you can start using OpenSpecimen if you want. So those are the other things that you can, of course, configure, which we'll be seeing in the next few slides. First and foremost, since I mentioned before, the data that you capture in OpenSpecimen can be sensitive, and you might want to control who can access what in the system. So when you create a user in OpenSpecimen, you can give them different levels of access, which we call as a user role, and you can also control what they can do in the system. So this is all controlled in OpenSpecimen by the admins. And the system is highly configurable. What I mean by this is not just the data fields that you capture, even the screens that is visible, how you capture the data per study, they can be configured as well. So again, we'll see some examples of it. So you can do different types of collections. First and foremost, we'll see how you can do longitudinal collections. So lot of times when you collect the specimens, it's for a specific study. It might be like, a specific lung cancer research, a diabetes study, and so on. So in these kind of study collections, you know what you want to collect, you know when you have to collect, how the specimens have to be processed. So it's a very predefined or well-defined process for the whole collection, processing, and so on. So now, in order to do this, what OpenSpecimen allows you to do is, first and foremost, it allows you to predefine the study with different details like time points, what what kind of specimens you want to collect. All that can be predefined. And once you have predefined, now the study coordinators or the nurse at the hospital can enter the patient data, accession the specimens, and so on. Once this is done, then it can be sent to the lab for processing, where they do it as per the SOP and enter the data along with the storage information. So let's look at this use case first. So for this, I'll be using our demo site. So we have a public demo site. It's demo.openspecimen.org. Anybody can sign up for an account. And we always have the latest version on the demo site, so you can access and try out the application. Right now, I'm on the home screen and have logged in as an administrator. So as I said, there are different levels of access. So when I log in, I see a menu like this, through which I can go to different parts of the application, as well as the home screen allows me to navigate to different parts of the application. So again, what you see in the system is dependent on what role you have. And also, OpenSpecimen is a web-based application, so you will be able to access it using any standard browser. So now, let me go inside a particular study. So in OpenSpecimen, we have a concept called collection protocol. So this is used for capturing or categorizing the specimens based on the study for which you're collecting, or it might be general biobanking. So you can create different protocols to capture why you're collecting the specimens. So right now I'm inside a longitudinal study. So you can imagine this is a study where you're collecting samples for specific research needs. It might be for a specific PI or a coordinators group and so on. When I go inside the study, I see a list of patients. These are all the patients that are already registered. And again, these patients can either be entered directly in OpenSpecimen, or they can be integrated and pulled from other systems. Like if you're using RedCap, Epic, and so on, they can be pulled from there as well. Again, entering patient data is optional. If you don't want to enter patient identifiers, if you want to just have subject IDs, all those are configurable. So when I say configurable, all this is configurable per study. Okay, so now let me go ahead and add a new patient. So this is the patient registration screen. So these are some basic demographic details. If you want, you can hide certain fields. Suppose you don't want to enter name, birth date, you can hide details. And you can also add new fields to a study. So when I say add new fields, so these fields that you see here, these are not part of the OpenSpecimen default data model, but we have a feature where you can add new fields as well. So let me go ahead and enter a example patient over here. So I'll skip some of the things. So most of these fields are optional. And either I can register the participant as immediately, or I can proceed to collection. I'm just going to register as of now. So once I register the participant, so that patient details will be shown as an overview. If you see, and there is also a unique ID that gets generated. So this is what we call as the patient participant protocol ID. You can either auto-generate it, or you can enter it, or you can again pull it from other system, which is a subject ID, maybe in RedCap and so on. Now, once you have registered the participant or patient, so you have an option also to enter consent data. So this is again optional. If you want, you can register consent. This is an example of a consent form that you can fill up, whether the patient said yes, no, withdrawn, and so on. You can enter these details. You can upload a signed consent form if you want and save this information. Again, this information, if you have already entered in Epic, RedCap, it can be easily pulled over as well. So that is the second step you can do. You can add many more details to the patient. If you have clinical data, it can be entered using what is called as forms. These forms can be attached at different level, like participant, visit, specimen, and so on. These forms are created using a form builder. So there is a form builder in OpenSpecimen. Through the user interface, you can add new forms as well. So I'll skip this for now. Let me go to the patient. And what I'll do now is go and collect the specimens. So right now, what I'll do is only collect the primary samples. So this you can imagine that as a clinician or a study coordinator who's registering the participant, accessioning the specimen, so they can enter the primary samples. Now, if you see that I'm entering the data for a pre-surgical event, so like this, you can have multiple time points within the study. So if you imagine a longitudinal study where you might have a screening visit, you might have a baseline, 30-day follow-up, 60-day follow-up. So these kind of time points or visits can be predefined in your study, and you can collect the samples based on that study definition. So, and within each of these time point, what needs to be collected, how it has to be processed, these can be also predefined. So now it is telling me that for the pre-surgical event, I need to collect these samples. I can either say, okay, I didn't collect urine, so I'm going to say this is a missed collection, and I'm going to collect the blood samples. I can enter other details like collection date, receive date, which hospital it got collected from. If I want to print labels for my samples, I can do that as well. And I can add additional details. For example, I can enter the centrifugation details for the blood samples if I want. If I'm freezing, I can enter the freeze details. So now I'd go ahead and enter the details. So now if you see under my visits for this patient, it is saying that the pre-surgical event has occurred, but there are other pending visits. So if you see there is a surgery, post-surgery, follow-up. So all these are predefined in the study. So you can create a study with all these details. That is what you're going to collect, how you're going to process. You can all define it in advance. So now once I have done this, if you see, this is a missed collection. So it is showing me in a different color. The collected samples are in green. And this orange means they're yet to be processed. So it is still in pending status. If you mouse over, you'll see the status. So now I log in as a technician, I receive the samples, and now I'm going to process the child specimen. So if I want now, I can process the plasmas and serums, collect them. And if you see now, each of the IDs that you see here, they are all auto-generated. So even every aliquot gets a unique ID, and this can be auto-generated with any format you want. But of course, if you have pre-barcoded tubes, you can also scan in those labels. And if you see even the box locations, they are auto-allocated if you want. So for example, the plasmas are going in box 1, serums are going in box 2. But if you don't want it to be auto-allocated, you can also do manual location, storage location as well. So I'll go ahead and collect or process these samples. And if I want to enter the freeze time, I can do that as well and submit. So at this point of time, what I did was I collected all the samples for pre-surgical event for this patient. And these are the details. That is urine I could not collect, so I marked it as missed. Then I collected blood, which I processed into plasmas and aliquots. Then the serum tube, which I again processed into serum aliquots. So this complete hierarchy shows you what are all the specimens you collected, how did you process it, So there's the parent-child relation that is maintained. And when you go inside the specimen, it will give you more details about the samples itself. So there are many fields that you can capture at each specimen level. Again, each of these pages that you see, that is starting from the patient list, the patient overview page, the visit page, the specimen page, these are all configurable per study basis. So based on what kind of data fields you plan to collect for your study, you can configure. If you want to hide certain fields, add new fields, you can do that as well. So now if I go back to my patient list, let me go into another patient here. And I also have a surgery event where I have collected some tissue samples. Now, suppose I have sent this FFP sample for pathology review and I get back the results. I can enter that as well. So which again is through the forms. So if you see there are multiple forms even at the specimen level, I can go ahead and add them like this. So this form that you see right now, these fields are not part of OpenSpecimen default data model, but we have created this using the forms module. So you can add new fields through the user interface easily as an administrator, and you can capture any kind of additional annotations like pathology annotations, clinical annotations through the and capture it at either patient level or specimen level. And some of this information, like especially the clinical data, if you already have it in other systems like RedCap, Epic, they can also be easily pulled over. So this is, in general, the whole process of, how you can predefine a study with time points. You can then let the patient registration happen either directly into OpenSpecimen or from RedCap or Epic. And then you can accession the specimens and then send it to the lab where they will process it according to the SOP, record any deviations if there was, like, for example, you might have defined that you want to make 5 serum aliquots, 5 plasma aliquots, but you only got, you got lesser amount of blood. So you will be able to, record deviations, which means that you enter lesser number of aliquots. You can enter a reason for deviation and so on. So this is in general the process for longitudinal collection. But you might have other type of collections, that is, you don't have a predefined clinical study for which you're collecting samples. It might be general biobanking. So you only know that you're going to get these samples on a daily basis, might be from multiple sites. You can just collect and process as you receive. In these cases, you don't have to predefine those study definition. You can just have a general biobanking protocol and collect the samples as you go. So these are different types of collection. And since I talked about configurability, I wanted to just show you one more example, just to show you to what level these studies can be configured. Let me see if I have an animal study. Yeah. So here, if you see, just to show you an example, there is an animal study. So if you want to do non-human sample collections, it might be mouse samples, it might be any other samples. You can do it within the same instance. So you can have each protocol or each study look completely different, right? So here, if you see, this is an animal study, and if you compare it to the human study, the list of fields are completely different. And when I try to add a participant, so instead of having name, birth date, gender, the age, and so on, so you have here, species, you have weight, you have color, which you didn't have in the human study. So this is again just to show you an example of how each study can look different, and this is all configurable, and this can be done from again user interface. You don't have to have you don't have to do coding or change the source code to do these configuration changes. Yeah. So other part is going back to my protocol. Let me also show you again about the storage location. So I talked about how you can enter the freezer locations. So here, if I go to this Alicot, I have put this specimen in a particular freezer location, freezer 1, rack 3, box 2. And in order to configure these, we have a containers module where you can create a freezer. You can create a freezer like a hierarchy, like it can have multiple shelves, racks, drawers, boxes, and so on. And each box can have different types of specimens. It can have different dimensions, like it can be a 9 by 9, 10 by 10, and so on. So here, if you see, it's like as if you open the physical freezer. So when you walk into the repository or your lab where you have freezers and you open the freezer, how will it look like? It will have 5 shelves or 4 shelves, which will have racks. And when you open the rack, or you open up the drawers, you'll have multiple boxes within it. And within the box, now when you click on it, it will show you the samples. So here, if you see, each box will have different test list of specimens, and they have different color coding as well. So just to give you a visualization of what kind of samples are stored within that box. So this particular box has all kinds of specimens like serum, urine, whole blood. So you can see that it's a mix of things. And if I go to this containers list, here you'll see the list of all the freezers that are there. Since I have logged in as a admin, I'm seeing containers of different sites, but you would be able to see it for a specific site as well. So every user can be associated to a specific site, and based on them, that they'll get access to data of their own site. So site, when in OpenSpecimen, means anywhere a sample is handled. So it can be the hospital, it can be the laboratory, it can be a site where you're sending the samples to. So every user will belong to a particular site. So here, this is how different freezers are visible. It will show you how much it is full, what is the capacity, and I can go inside any freezer and see the details of it as well. Let me see if I can show you one more freezer here. Yeah, so here it's kind of like a tank and you have boxes and so on. So you can have different hierarchy, different structure of the freezer. So every freezer can look different. It can be an LN2 tank, -80 freezer, and so on. So yeah, so now going back, so this is how you can enter the samples, and you will be able to get the data out using the reporting tool. So in OpenSpecimen, we have a very powerful reporting tool which allows you to look for specimens of interest. So this might be, as an end user or a tissue banker, you might want to look for the specimens. So you might want to use the reporting tool, or it might be for the researcher who is requesting for the specimens. So as a end user, you can apply filters, which means that you add conditions to look for specimens on any field of OpenSpecimen, including the custom field. Then you configure the results view the data, and from there you can do multiple other things. So we'll look at one of the examples. For example, I'm, as a researcher, interested in looking for female gender patients, and I'm doing research on breast cancer. So I'm looking for a tumor breast tissue with specific diagnosis. So how do I do that? So we have the queries module for that, which is our reporting tool. So here you can create any number of queries. You can save those queries so that you can share it with others, or you can frequently use the same query, then you can save it. In order to create a new query, you go here and you can add filters from here. So here you can either look for specimens from a specific study or multiple studies. I'll look for a demo longitudinal study. And here I'm going to add some conditions. So I'm going to look for, say, tissue specimens. So I'm going to add some conditions over here. This is called the filter. I'm going to add two values over here just to show you how it will look like. So this means that I'm looking for specimens which are either frozen tissue or FFPE. Then I'm going to add the anatomic site, which is breast tissue. And I also want to make sure that they are a tumor tissue. So I'm going to add the pathological status. So as long as you have captured these details, you will be able to do queries or reports based on it. Now I want to also make sure that this is from only female gender. So I'm going to add that also as a condition. So like this, I can add any number of conditions. And as I've added my conditions, I can see if I am getting any results. So there are 40 patients which are matching all these conditions. And I also want to add filter on diagnosis. So I'm just going to add that as a parameterized filter. I'm going to show you what that means. So now I'll go ahead and view records. So then now this is giving me all the female gender patients having that specific type of tissue sample that I have added as filters. Now, since I made diagnosis as a parameterized filter, it appears on the results view. So I can dynamically add that filter and look for specimens. For example, I want specimens only for this diagnosis. I can just select it and it will further, drill down to those set of results. Now the other thing I can do is, so that you see that it is showing me very specific columns. What if I want to see more data? Then I need to go to columns. I can configure whatever I want to see. Suppose I'm not interested in this, but I'm interested to see where they are in my freezer. So generally, when I get requests for my samples and I want to take it out of the freezer, I want to know where they're stored. So I'm going to include the container details. I already have the diagnosis and some participant details. I'm just going to remove race and say done. So now the results will be refreshed based on what columns I included. So this way I can include any number of columns, even if you want to include clinical data, pathology data, the forms that you have filled in, either directly into OpenSpecimen or from RedCap. So those can be easily queried on and also included in the results. As you see, you'll see now the container details as well. That is where they're stored. The anatomic side, they're all breast tissue samples, the specimen IDs, the diagnosis, and every other detail that you need. Yeah, so like this, you would be able to do reporting based on any field, add filters, look for the specimens, and then you can also save these reports. So search for female patients with tissue samples. Yeah, you can save this so that if you're doing this query repeatedly, then you don't have to recreate that every time. So this will come on my dashboard. So when I save it, it is only for myself, but I can share it with others. I can assign it to a folder and share it with others. And I can even schedule the queries. So suppose you want to schedule it to run on a monthly basis, quarterly basis, automatically, and send reports to someone, you can schedule it as well. So you have the option of schedule over here. So you can automatically run this reports and notify users when it's ready to download. So these are some of the features of query as such. So you can export the results as well as a comma separated value files. You can save them, schedule them, and so on. So the other type of report is what we call as count-based report. Suppose you're not interested to see the actual data, but you just want to see counts of specimens. So you can do that as well. Let me just see if I have an example that I can show. So what this will do is it will not show you the actual data, but it will show you the counts. Sorry, let me just log in as a different user. So here, if you see, this is an example of count-based report where I'm able to see different specimens by just the count. Yeah. So this is an example of a summary report or count-based report. So you can either do the raw data-based report, which we saw earlier, or these kind of pivot table kind of reports that you can do. And these can as well be scheduled. You can export this into a CSV and so on. So the reporting tool is very robust and it has a lot of different functionalities. What we saw right now is a very small percentage of what you can do through the queries. So let's move ahead with others. So again, now once you have the data and if you want to publish it to your researchers where they can request the samples and you want to distribute the specimens, that can be done through what is called as the catalogs. So the process is you can publish your data either without login to the researchers where they can submit a request, or it can be a private catalog which needs login. And as the researchers, they can look for specimens of interest and submit a request. And once you have the request, there are some users called as catalog managers. So when you create a catalog, you can create managers who can approve and reject the request. And finally, the technicians or the biobank users, they can process the request and send it to the researcher. So here, you can have any number of catalogs. So we'll see this particular catalog. So the catalog has two parts to it. One is what we call as the dashboard. So the dashboard can have different graphs like this. And these graphs are also created using the reporting tool. So you can create different summary-based catalogs or summary-based dashboards using the reporting tool. So this will give you this will give the researcher a brief idea about what kind of data that you have in your biobank. And these catalogs can also be embedded in your biobank websites. So now as a researcher, I'll go inside the catalog. So the catalog is nothing but a very simplified version of the reporting tool we saw earlier. If you remember in the reporting tool, there were hundreds of fields which are present in OpenSpecimen. So you can have a smaller version of it, that is, you can expose very limited fields to the researchers so that it does not confuse them. And you can have multiple catalogs. So it can be a study-based catalog where you only publish the data and data fields which is specific to that study. So now as a researcher, okay, I'll say I'm interested only in alive patients. I'm interested in specific, race, type of specimens. Maybe I'm interested only in DNA samples. So I can keep adding conditions like this. And as you see, there are very limited filters that I see, and these are all part of configuration of the catalog. I can either see the list like this as a card view or as a list view. Now, once I have the specimens of interest, I can select a few specimens, add to cart like a pick list. And from the cart, I submit the request. So here is where I'll submit the request saying that I'm interested in these specimens. So as a researcher, I can myself look for specimens and request. Usually what happens, the researchers give you a phone call or email you asking for, do you have such specimens? Instead, if you publish the data to the researchers, they can log in, submit the request themselves. So I'm just going to enter some details over here. Okay. And submit the request. So there's a request created. Now what happens is for the biobank users who are the catalog managers, they get notified that there is a request. Now they can log in, go inside the catalog and view the request. So there's a request dashboard As you see, there are different status that is happening. So the request that came from the researcher, now I can go ahead and process it. So if you see here, whatever they filled in, I can see that. I can approve the request, notify someone. And after I approve the request, I can go ahead and distribute the specimens. So what I mean by distribute is I'm dispatching the specimens to the researcher as per the request. Or if I want to, remove certain specimens, I can only distribute few of them if I want, whatever I need, and I can distribute. When I distribute, I can give details about which project is it going to be utilized for, who is receiving it, which site, who was the requester, when am I, what is the date I'm distributing, any tracking URL, all those can be tracked. And then I'll say next. So this will show me the list of specimens. I can apply a cost if I want, and then go ahead and distribute. So now this got distributed. What this means is the specimens that were there in your repository, now you have processed that request and send it to the researcher. So now inside, if you go inside this specimen, the data will still exist in OpenSpecimen, but it would be marked as distributed. It's no more available. The quantity will become 0. The location will be blanked out. When I say location, it's the storage location. The freezer location would be freed up. And it creates something called as an order. So you know that you have a history of all the dispatch specimens. So orders is nothing but all the specimens that were sent previously to different researcher. So as an admin, I can track and see what all specimens were sent out, to whom was it sent, where was it sent to, how many specimens were sent. If you want to charge the researcher, you can create an invoice and so on. So this is how you can track the whole process of request, reviewing the request, and processing the request to distribute the specimens, so that you track how the specimens were utilized, and those specimens will be marked as no more available, so that it is removed from your storage location. If you have a use case where the researcher sends back those samples, you also have an option to return the specimens and store it back in your freezers. We do have option to mark the specimens as returned. So if you see, there is an option to return specimens as well. So that's the, what you saw till now, as I said, is the whole process of the specimen tracking. So you collect the specimen, you process them, you store them in your freezers, and finally, after storage, you let the researcher look for the specimens and you disperse or dispatch the specimen. Now, let's look at some of the additional features. We do have a module, what is called as supplies. This is more for tracking inventory. Again, this is an additional module, so it's an optional. If you don't want to use it, you don't have to. It allows you to track any inventory management, like tubes, kits, gloves, and so on. So, for example, if I go to that module, so initially when you purchase inventory like kits and tubes, you enter that in the inventory, and here you will mention how many of the specific supplies did you purchase, what is the procurement date, what is the expiry date, and so on. And as you're utilizing it, if it's a barcoded tube or a barcoded kit, it will automatically mark it as used. Or if it's just unbarcoded or non-barcoded supplies, you can also manually say how much was utilized. Yeah, so this is the supplies module where you can track the inventory. Recently, we have developed something called as a workflows module. What we saw previously, how you can enter samples, you can predefine studies and you can enter, but this is more elaborate. So you can also enter a lot of other details of your SOPs within this workflows module. Just to give you an idea, I'm just going to show you an example of the workflow. So you can have different workflows for, say, heparin tube processing, different DNA processing, blood processing like EDTA tube processing, and so on. So when I say blood processing, you'll see that you have all these steps. So you receive the primary tubes, you pull them, you then spin it, then you create, you dilute the EDTA tube, you then create the serums and plasmas, freeze them, and so on. So this is just an example, but what you see here is like kind of like a wizard-like data entry, step-by-step process of each step of your SOP. So you don't have to go to multiple pages or multiple places to enter the data. So you have a very well-defined SOP. If there are deviations, it will ask you to enter comments and so on. And you can have any number of such workflows tied to your protocols and enter. So this is again a different module, which if you have very elaborate SOPs and you want to track every step and data of your SOP, this is a good module to use. We have a feature for importing data in bulk. We use this extensively for legacy data migrations, but of course you can also use it for your daily data operations, especially for form data entry. I was talking about pathology review data. So you might receive pathology review for multiple tissue samples at a time. So you don't want to enter it one by one. So you can enter it in bulk. So we have what we call as bulk data entry or bulk import. For that, what happens is when you go inside the bulk import, you'll be able to enter data in bulk for any data in OpenSpecimen, may it be patient data, consents, specimens, and so on. And when you want to enter the data, you need to first download a template file, which will have exactly the same fields that you see in the user interface. So it's like a CSV, empty CSV file with only the headers. And then you populate that data file and upload it back. So that is what you can do, the bulk import. Again, as I mentioned, we use it for the legacy migration. What it means is, suppose you decide to use OpenSpecimen today, but you have a lot of data in Excel sheets, Access database, and so on. So now if you want to transfer that older legacy data from your different database or files into OpenSpecimen, this feature can be used as well. So that's the enter data in bulk. We do have a mobile tablet app where you can enter data, whatever we saw, like patient registration, clinical data, specimen data, you can enter through the mobile app. One of the main advantage of this mobile app is you can also do offline data entry. So in case you're going off-site where you don't have access to internet, so you can enter data and then you can come back to network and then sync the data. So that's one of the other modules. We saw very basic module of consents when we were seeing the demo of data entry, but we do have a very elaborate e-consenting module as well. If you want the patient themselves to enter consents, or if you want to have mimic your exact consent form, like in OpenSpecimen, enter the data for it, send reminders to enter the consents, any forms from patient. So there are multiple features again with eConsents. This is again a separate module available for use. So every data in OpenSpecimen that you enter is audited. So right from login, add, edit, delete, query, everything that you do in this application, they're all audited. So you'll be able to generate reports based on that. The system is based on 100% REST API. So whatever you saw today using the user interface, they're all also using REST API. So what this means is you can easily do integrations with different application instruments, because you can call all these add, edit APIs, which you can utilize and integrate with other systems. So some examples of integrations that we have done is RedCap, OpenClinica, where you want to pull some clinical data from RedCap to OpenSpecimen, you can use this REST APIs. We have plugins, what we call as plugins. to get the data from these different systems. We have also done integrations with instruments like automated freezers, like Hamilton BIOS, liquid handling systems, like Easy Blood. So whenever you make aliquots, whatever file it outputs, you'll be able to import into OpenSpecimen to create those aliquots automatically. So these are some of the examples, but similarly, you can have any other integrations that you need, you want to do with OpenSpecimen. So now we saw so many features. All these we have been developing since 2009, and we are continuously improving. So we make releases every 3 months because we are working with so many different clients, different use cases. We are adding new clients every now and then. So they have multiple use cases. So every new release has bug fixes, enhancements, usability enhancements, all this based on the community feedback. So, and again, when we say we make new release every 3 months, it doesn't mean that you have to upgrade to every new version. You will be able to choose which version you want to upgrade to. We have release notes which will give you details about what is there in that release, and you'll be able to choose what you want to upgrade. And with every release, we make sure that we do performance testing, scalability testing, because a lot of our clients have million+ samples. A lot of clients have hundreds of users using the system concurrently. So to make sure none of these cause performance issues, we give very high priority for performance testing and fixing any issues if reported. So those were some of the features and the highlights of OpenSpecimens as such. There are many other things which you can learn. We have a public help documentation. So if you go to help.openspecimen.org, this is one of the very important thing that you can use when you're doing evaluation. So here you see that you have the user manual, you have the deployment guide, and everything that you need. Especially in the user manual, we have step-by-step procedure how to use each feature of OpenSpecimen. Not only the textual part, you also have some training videos where you can watch and learn OpenSpecimen. So that's one of the things that you can use. We also have a public forums that you can subscribe to. We give we do posts about OpenSpecimen, new features, about such webinars and so on. So we highly recommend that you subscribe to it. The system that I was using today, this site, this is our public demo site. So demo.openspecimen.org. You can sign up if you want to try out the system. And finally, if you have additional questions, if you want a very customized demo, you want to look at OpenSpecimen, you can contact us at this email. So that was it. Let me now see if there are any questions. So yeah, so there are a few questions. There is a question: how do you know they will use the samples in a way that is consistent with exec review consent? Is there a material transfer agreement process that is built in? So one is when you want to use the samples, you can have fields to upload MTA documents if you want. The other is you can also validate consent. So there is a feature where when you want to distribute specimen, you can put a validation of consents, which will restrict you from distributing the specimens if it is not as per the consents. So there are certain validations that you can set so that the distribution does not happen, it does not happen against those as such. So the other question is, can OpenSpecimen be integrated with Encore? Yes, it can be done. In fact, one of our clients, Emory, has done some basic integration as well. The next question is, how would you document adding something to a derived aliquot? For example, if you collect viable cells from whole blood and freeze with a cryopreservation solution. Where could you include that you added the cryopreservation solution to the cells? Yes, there are multiple fields. When I showed, let me just show you the specimen page. Yeah, so this is, for example, the specimen page as such. And if I go to the parent specimen, so these are some of the fields. And you also have what is called as collection container, where you can put the details about if you added any additive and so on. So what you see here are what are present as default fields of OpenSpecimen. But if you want to add any additional fields, we do have a feature called custom forms, where you can add any number of fields. As you see, you can add all these details, and you can add additional fields if you want to capture details about cryopreservation or any other details. You'll be able to add those fields and enter the details. Okay, there is another question from Michael. Have several questions. Okay, I'll try to answer what I can. Required fields are not marked with asterisk. It is marked, and if it is, so let me just show you actually this page. Yeah, so if you see here, it marks the specimen fields as mandatory if it has a mandatory field. And printing labels, can you show us customization of the format? So the way printing works in OpenSpecimen is you can define what is called as print rules, which will define what is the content of the label. And then you need to have a printer software. Generally, our clients use Bartender. That is where you'll be customizing the format of the label. So OpenSpecimen, when you say print specimen, so there is a print here. So now when I say print and submit, this will send a file which will have the contents of the label, and it will write it to a folder which will be picked up by your printer software like Bartender, and that will send it to your printer. So it is automated, but you need to have an intermediate software for formatting the label and sending it to your network printer. Is there integration with Zebra printer? As I said, what kind of printer does not matter to OpenSpecimen, because it's going to be via the printer software. So the printer software is the one which will be directly talking to your printer. What do you use for PDF reporting? Something like Word. So we don't have PDF reporting. The reporting allows you to export as a CSV. We do have some places like in collection and order report, you can download a report in a PDF format, which we call as manifest. But other than that, the reporting query results can be exported only as CSV file. Okay. Let me just see. I think I got lost a bit. Sorry. Sorry to clarify, does not get added to the parents. Is there a way to add directly to valid code? Yeah. So there's a follow-up question about adding data to a specimen. Yes, you can add it to a parent specimen as well as directly to a Lalicot. You can add data to any level. So there is no restriction on that. Does the eConsent form support multi-language? Yes, definitely it does. So you can have, so if you go to eConsents on our help manuals, so you'll see certain, there is also a video. And as you see, it supports, these are all the features of eConsent. It supports HTML formatting. You can have versioning, you can have multiple languages, validations, and so on. Okay, are there any other questions? Sorry, I missed one more. There is a generating PDF for eConsent. Yes, that is allowed. You can generate the eConsent as a PDF as well. Yeah, so within the e-consenting module, you can also send patient surveys, not just the consent forms. You can send surveys to the patient as well. So you can, you will be able to send notifications, send reminders so that the patient can enter the survey questionnaires, consent form offline and send it back. How much does it cost? You can contact us. I'm not sure what cost you need. Is it for e-consent or for the software itself? We'll get back to you over email on this. Do you have an integration with IRB system? Not right now. We have not done any such integration right now. But as I said, as long as the other system can provide us data in any format using the REST API, it generally can be integrated with any system easily. Okay, any other questions? Last minute questions? Last call. What type of programmer support does each site need to adopt? We You don't require any programming support if you want to just use OpenSpecimen. And you're not going to develop any plugins or integration plugins within your institution. For all the configurations and all the features that we saw today, like creating studies, creating freezers, configurations of the screens, none of these need any programming skills. So the programming skills is only required if you want to do additional customizations or plugins or to integrate with other systems. And of course, we provide services if you need those kind of things. Okay, that's it then. I think those were the questions. As I said, please, you can feel free to contact us at this email address, or if you have general questions, you can ask on the forums. So if you want a specific demo, we'll be happy to do that as well. And that's it for today then. Thanks everyone for joining. Have a great day or evening wherever you are. See you in the next webinar. Thank you, everyone.